I'm Kathy Conley, also a graduate of the 2014 EMBA class. And a little bit about Linda. So Dr. Linda Vahdat is a professor of medicine and director of the breast cancer research program at Weill Cornell Medical College. Her expertise is in breast cancer. She's involved in conceptualization, design, and conduct of phase 1 through phase 3 clinical trials, mostly in highly adjunctive and metastatic breast cancer. Her major research interests include strategies to understand and prevent metastases, and especially in difficult-to-treat breast cancer called triple-negative breast cancer. Linda's here today to discuss disruptive approaches to preventing metastases in breast cancer. She can say this stuff better than I can, by the way. And how the value of new drug development and cancer therapies can reach everyone, most importantly, the patients. So a couple of things that aren't on paper. One thing that I just learned a few minutes ago is that in 2013, Linda won an Emmy for a special health and science program entitled Breast Cancer, New Options and Promises. So she's world famous for multiple reasons. And as a former colleague in the class of 2014, I can tell you that Linda works with great passion and compassion, and it was a pleasure to be in school with her over the past couple of years. So with that, I'll turn it over to Linda.
Wow, what an introduction. And you'd think by this time I know how to use these things, right? But it says it's on. Can you hear me? No? Hello? 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 Oh, okay. So obviously I'm incompetent on that part. Okay, so it's a pleasure to be here. I'm trying to remember the last class I had in here. Maybe it was Analytics Edge. And I think Dr. Bersimas was talking about trying to pick chemotherapy for gastric cancer based on an analytic algorithm that he had developed. Very interesting, and actually we're going to start to apply that to breast cancer. But I'm very happy to be here to talk to you about disruptive approaches to cancer therapy. And I just wanted to say that the word disruptive is not a word which is frequently heard in my world. In fact, when I use that word disruptive, my colleagues knowingly look sneaky glances to each other saying, oh, it's more of that MIT stuff. But I would argue that you have to be disruptive in cancer in order to get it to the next level. And with that, I'm going to explain to you some of the things that I think that we're doing actually that are disruptive and will get us to the next level. So cancer is a complicated beast. Bob Weinberg, who is from MIT, attempted to put some structure around it, specifically so that we could begin to target therapies towards those 6 core capabilities of cancer. When you look at this, hallmarks of cancer, which is what it's called, there are certain things which sort of pop out about cancer, which maybe you don't see in normal cells. Certainly that that signaling is ad infinitum. There's no cell death. Tumor cells know how to evade growth suppressors, and also tumors can avoid immune cells. They can have to be able to get places. And it was actually felt that if you targeted all of these core capabilities, that perhaps we could begin to crack the nut that's known as cancer. Well, it's a little bit more complicated than that, of course, because I said it's complicated. But this has actually turned out to be a sustainable framework on which to base drug development. And looking at some of the core capabilities, which have since been expanded, there are a number of targeted agents, which we've been using actually for close to a decade. So, for example, the EGFR inhibitors up here, these are drugs like erlotinib, cetuximab, a number of other drugs which target a lot of different solid tumors, and they have been shown to improve survival. I really like the PARP inhibitors because the PARP inhibitors actually target BRCA1 and BRCA2, which are genes which are predisposing mutations in breast cancer. VEGF inhibitors of VEGF signaling are drugs like bevacizumab or Avastin, and there are a number of oral VEGF inhibitors, which are the standard in a number of solid tumors. Telomerase inhibitors are sort of interesting, and they are still in development. And cyclin-dependent kinase inhibitors are sort of the new kid on the block in breast cancer, with a drug called palbociclib just recently approved in breast cancer, which makes hormonal therapy work better. So it wasn't a bad idea to sort of put these framework together on which we could then have a very sort of organized way to approach how we treat cancer. But I would say that one of the most exciting areas in oncology today, and I'll say, disclaimer, in the beginning I was a total disbeliever, was this whole issue of how do cells avoid immune destruction. And specifically with that, we're going to talk just a little bit about the checkpoint inhibitors, because I think if I didn't even give you a little bit of insight into that entire area, I would be doing you all a disservice, because I think this is probably going to go places. But there are issues. So as you know, I'm an oncologist, and every year, end of May, beginning of June, there is a huge meeting called the American Society of Clinical Oncology, or ASCO annual meeting. And basically 30,000 of your closest friends who are oncologists, pharma, research scientists convene recently in Chicago in order to share ideas, to share trial results, and to sort of set up the whole next set of research collaborations for the future. But what's important about that meeting is that if things are presented at ASCO, especially in a prominent position, usually there are things that are going to change the standard of care. And we're always looking to change the standard of care, specifically march towards a cure in cancer. So I would say that absolutely the hottest topic at ASCO this year was all of the data surrounding the checkpoint inhibitors. And checkpoint inhibitors is just a way to describe the drugs which sort of modulate the immune system. So when we talk about checkpoint inhibitors, we talk about the PD-1 pathway and the PD-1 ligand. And sort of the way it works is that you have an immune cell called a T cell here, and the T cell's job is basically to find distant invaders and to sort of knock them out. But the thing is, sometimes T cells are not that smart, and sometimes they can look at you or cell and they can attack you, and that's really bad because then you can get really sick. T cells usually respond to antigens, and usually they have to talk to a tumor cell. So a tumor cell has an antigen that the T cell recognizes. Then you can have activation of the T cell, and you have this army of T cells that will come and they will sort of wipe out a tumor cell. But the thing to have to remember about tumor cells are the big co-opters. And what they do here is that basically what they do is that they co-opt the immune system. They sort of, they put it to sleep. And the way that they do that is that they have something called PDL1 or PDL2. And this is sort of the checkpoint here, this PD-1 receptor here. And what it does is it gets totally neutralized. So the tumor cells co-opt the immune system, so the immune system gets put to sleep. So tumor cells can just go and do whatever they want. And what's really interesting is now there are drugs which are PD-1 and PDL1 inhibitors. So basically the thing is, if you can disrupt this sort of communication here, then all of a sudden the T cell wakes up, it can see the tumor, it can fight it. So all of a sudden you have your immune system fighting the tumor. So in oncology at ASCO, if you have something called a plenary talk, that means you're a big dog. It's like the top 5 abstracts get to be presented. And there was a big plenary talk given by Jed Walchuk from Memorial on drugs in metastatic melanoma. And just to sort of put it into context, melanoma has been one of those diseases where nothing works. And when I say nothing, I mean nothing. Nothing happened in that disease probably for at least 25 years. And then all of a sudden, the immuno-oncology came along, specifically ipilimumab, which is a checkpoint inhibitor, and all of a sudden you had actually the tumor shrinking, which never happened, and you actually had some people living for more than 5 years, which never happened. So the issue is always, well, you got one drug, how does it compare to some of the other checkpoint inhibitors that we use? And so there was this trial that was presented in patients who had melanoma that had spread, and they looked actually at 3 different combinations. They looked at ipilimumab, which is a drug which was approved several years ago for melanoma. Another drug called nivolumab, which is another checkpoint inhibitor, or the combination of both. And lo and behold, everyone was so happy because they said, wow, look, Ipi, 3 months. And remember, this represents an improvement. The average time it kept the cancer under control, which in our world we call progression-free survival, or PFS, was 2.9 months for the Ipi, 6.9 months for the Nivo. So okay, good, Nivo's better than Ipi. But the combination, 12 months, which of course is pretty big in oncology. So we were all happy about that. But then what we found was that what price tag does this come? Because there's a whole big movement in ASCO for last year and this year about value and cost. And so what they saw that for Nivo and Ipi, that it would cost close to $300,000 for 1 year of therapy, about half of that for some of the monotherapies. So that has brought a very interesting discussion around this concept, because Len Saltz, who gave one of the discussions after this about the issue of value and benefit, actually said, well, gee, you know, this is a good drug, obviously. It works in a lot of people. And if we were to give 1 year of combined therapy to people, how much would that cost us, right? And it would actually come out to $174 billion a year just to treat patients with metastatic disease for the first year only. So this is a discussion that we've never had in oncology before. So that in and of itself is disruptive. So that brings us, and now we're at MIT, so of course we have to have a pseudo equation here. Does the benefit equal the value? And furthermore, it was posited, how do you measure that value? Does it include cost and toxicity? And I will tell you, in oncology, the concept of even talking about this is as disruptive as the idea of a checkpoint inhibitor. And so it's going to be interesting to see what moves forward. So I don't want you to take home that, oh, these drugs are really expensive. Yes, we have to talk about that. But what I want you to take home is that when they work really, really great. And they work, it seems, in about 20% of pretty much every solid tumor. So the onus is on us to really identify what we call a biomarker in order to really figure that out. So is immuno-oncology disruptive? Absolutely. It's innovative, it's groundbreaking, and it's troublesome. And it's troublesome from the value cost-benefit analysis. Okay, so now what we're going to talk about is some of my research, and I'm really excited about this stuff. This is data that we just presented at the oncology meetings on Monday. And basically what we're looking to do is that we are looking at strategies that target what we call the tumor microenvironment. And I mean, I'm throwing around a lot of these words, and I really want to just define what the tumor microenvironment is. The tumor microenvironment is the ecosystem of the tumor. It's all the things that allow it to grow, to spread, to be, to exist. And you can see all those things right there. And it's the tumor microenvironment, which I believe, and our group at Cornell believes, is really what we should be targeting, in addition perhaps to the tumor itself, but it's the microenvironment. So what is our current strategy to target tumors? Well, we target the tumor itself. We target the primary tumor, we target the metastasis, we slice it, we dice it, we stain it, we do fluorescent in situ hybridization, we send our specimens off to precision medicine, we send them to Foundation Medicine up the street, but we're always totally geared on the actual tumor itself. And so I would argue that perhaps we should not be doing that, and perhaps we should be looking at the infrastructure that the tumor utilizes, because if we can pull the infrastructure out, perhaps a tumor cannot get from point A to point B. And this whole concept of pulling out the infrastructure has been likened to having a car that has no gas, or because we're at MIT, we have to talk about electricity in the battery, but it's exactly that. So you have a tumor that has nowhere to go. That, you certainly could say, is a way to maintain tumor dormancy, perhaps. And if you can maintain tumor dormancy, if it never bothers you, isn't that a cure? And I want you to think about that. So what I'm going to do is I'm going to show you the ways that we believe we can affect the tumor microenvironment. And as you all know, I'm a breast cancer doctor. And I've always been very interested to understand why tumors spread, because I've always dealt with patients who had metastatic disease or who had tumors that were at high risk of tumor coming back. So my fellow, I was very proud of her, did a terrific job of presenting our data this past Monday. So breast cancer is a common disease. Over 1 million women a year are diagnosed with it. Over 400,000 women per year die of it. Most patients die from metastasis. And breast cancer is very different from other kinds of solid tumors in that it has what we call a long natural history. So you never tell a breast cancer patient, oh, doesn't come back in 5 years, you're okay. You talk about 10 years, you talk about 20 years. So there's this long periods of tumor dormancy. So where are these tumors hiding? So at Cornell, so our question was, why can tumors be dormant in a cult for years and years and years and then come back? So at Cornell, when I went there in 2002, I was very lucky that there was a whole group of scientists that formed a critical mass that wanted to understand and answer this question too. However, they looked at it from what was the contribution of cells from the bone marrow, which is sort of like a shopping center, your bone marrow houses all these things. What was the contribution of the bone marrow to tumor progression? And so many of the scientists that I worked with had spent a lot of time figuring out all the different steps. So for example, one of my colleagues, his name is Shaheen Rafi, he's a crazy Persian dude. He basically, brilliant, basically figured out that if you labeled bone marrow cells and then you looked at a tumor, you could see those labeled bone marrow cells in developing tumor neovasculature. Another guy that I worked with, David Leiden, said, well, gee, tumors, do they have homing mechanisms? Do they know where to go? And so basically they showed that no, tumors don't know where to go. What they do is that these cells from the bone marrow, those VEGFR1 positive cells, they come out and what they do is they leave a popcorn trail for tumor cells to follow. And then VEGFR2 EPC, so the tumor cells get to that distant metastatic site, and they're just sort of hanging out there. They don't come with suitcases that have their infrastructure. So what they do is they co-opt it, and they get these. And where do they start? Well, they got to create the highways. So what they do is they get these VEGFR2 positive EPCs, or endothelial progenitor cells, to put down blood vessels. So those are the superhighways to which all the infrastructure comes to them so that they can grow and spread. And so when we saw that, we said, wow, that's really interesting, but these are mice, right? It's not people. So we actually set out to look to see if the same thing happened in women with breast cancer. The bottom line is that it did. And so just to give you a sense as to what that might look like, and so this is not only in mice, but this is also in women with breast cancer, is that the primary tumor niche stimulates the bone marrow compartment. It releases these VEGFR1 positive cells that lead the popcorn trail for tumor cells to follow to a distant site, where it's formed what we call the premetastatic niche. So there's no premetastatic niche. Then what happens is that that forms what we call avascular micrometastasis. But they need infrastructure. So what happens is that these VEGFR2 positive cells come out of the bone marrow niche, they go to the premetastatic niche, and then they flip the angiogenic switch, transitioning cells from being avascular and micrometastatic to being vascular and macrometastatic. So what we said was, is there any way to stop that process? And if we stop that process, that means that people won't relapse. And so that's how we started to look for drugs that might actually do that. And that's where our clinical trial came in. And that's actually where copper came in. So copper is relevant in multiple biologic processes which are important in cancer progression. Copper is also critical to help cells move. So when cells move, what happens is that copper goes from the nucleus, or the brains of the cell, to an extending filopodia, which is sort of like a foot, to help it move. It's critical for cell migration. And actually, there's been a lot of research recently showing that it's really important for what we call oncogenic signaling in particular models, and BRAF. BRAF is important in melanoma. So there are a lot, and this is just the tip of the iceberg of all the things that copper is really critical for in the biology of tumorigenesis. Tetrathymolipdate is a drug which is used for people who have an inherited genetic disorder called Wilson's disease. And so many people have looked at that in mice, and they showed it prevented tumors from growing in mice. They actually gave it to people who had a lump of cancer that you could see, and it didn't really shrink it down. So the way we used to think was that if it didn't shrink it down, well, it's no good, we're not even going to bother using it. But we thought that there was a value to it, and what made us think there was a value to it was this experiment that was done at the University of Michigan, in which they had this mouse model, and you see him talk a lot about mice. I'm really a people doctor, but you can do a lot of things to mice that you can't do to people. So we had these mice, and these mice are genetically programmed to have lung metastasis when they're 205 days old. And so what they did was that they gave these little mice, half of them they gave them the regular diet with water, and they gave the other half they gave TM, this copper depletion compound. And what they saw was in the mice that got their regular diet water, as usual, what you would expect, they got their lung mets. But they found that the TM mice did not. But what was really striking was when they took TM out of their diet, on average 2 weeks later, they developed lung mets. And so that made us think that copper depletion was inhibiting the processes which were critical for tumor progression. And we thought that this actually could apply to our breast cancer patients. So we designed a study to test this hypothesis. So what we did was that we said, well, okay, we're going to test this hypothesis in women who have breast cancer that is at high risk of tumor coming back. We're going to give them TM orally, it's a pill, for 2 years to get them copper depleted, and copper depleted is just this CP target. And what we're going to do is we're going to look at these VEGFR2 positive EPCs. These are cells that flip the angiogenic switch. And we're going to look at a bunch of other biologic endpoints, and we're going to see what happens to these people. Do their tumors come back? And so just giving a little bit more granularity in the trial, and this may be too much information for you folks, but I'm going to give it to you anyway. Because you're all MIT, so you should be able to understand it. So what's high risk to us? Well, high risk to us is stage 3 and stage 4 NED breast cancer. So stage 3 means you got a lot of positive lymph nodes or a humongous tumor. A stage 4 NED breast cancer means, your cancer came back, but somehow you got rid of it. We don't care how you got rid of it. You could have cut it out. You could have radiated. You got a chemo. We don't care. All we care is that when we do imaging studies and we examine you, and we do every blood test known to man, we see no evidence of it. But we know your tumor is going to come back. It always does. Patients had to consider what was considered standard therapy. Now, we started this trial in 2007, and in 2009, we started to get some signals. And one of the signals that we got was that, well, gee, this might be particularly good and particularly hard to treat breast cancer called triple-negative breast cancer. The other signal, well, actually, it wasn't a signal, from a biologic point of view, became apparent because the field's moving forward, was that triple-negative breast cancer is very aggressive. And what we found was that a stage 2 triple-negative breast cancer had the same prognosis as a stage 3 non-triple-negative breast cancer. So being ever inclusive, we broadened our entry criteria to include the stage 2 triple-negative breast cancer patients. When we originally designed the trial, we were going to enroll 50 patients. Because we felt we got a signal in triple-negative, we increased the N to 80 to better study these patients. So patients were given oral daily TM. And these women are so dedicated. They came to visit us every 4 weeks. We even opened up a special TM clinic day for them to make it operationally good for them, where we examine them, we check labs, we do research bloods. In real time, we do something called flow cytometry, looking at those EPCs and those HPCs, these bone marrow-derived progenitor cells, and then we stick everything else in the freezer to look at it at a later date. We imaged patients every 6 months to make sure that they had no sign of their cancer coming back. It was an intent to treat population. We stratified by copper depletion and molecular subtype, and we looked at other outcomes, such as toxicity attributable to EM, time to progression, things like that. So just to give you a sense of the type of patients that we enrolled in this trial, at the end of the day, we enrolled 75. The median age was 51. 60% of patients were stage 2 or stage 3, and 40% were stage 4 NED. When patients were what we call an adjuvant, they had a stage 2 or 3, they had a median of 6 positive lymph nodes, which is actually quite high risk. For the patients who were stage 4 NED, 40% of them actually had visceral disease. So basically, disease had gone to an organ and they'd gotten rid of it. It wasn't just the lymph node that got popped out. And as we predicted, or as we designed it, about half the patients in the trial are triple-negative breast cancer. Now, I haven't talked a lot about triple-negative breast cancer, but suffice it to say that it is a very aggressive subtype of breast cancer. There are no targeted therapies yet, and there's a lot of research being done about it. So the first question always is, oh, you're giving the people these pills, this TM pill, can you copper deplete patients? And the answer is yes, we can. This is a plot that shows you that we can deplete patients with TM as measured by a copper carrying protein called ceruloplasmin, and it's sustained over time. We found that copper depletion was most effective in the triple-negative breast cancer patients, the dark line here, and we copper depleted them, 91% of them, by 4 weeks. So the primary endpoint of our study was what was our effect on these cells which are critical for the angiogenic switch, these VEGFR2 positive EPCs. And this actually represents over 2,000 data points. And the bottom line is that we were able to really drive down these cells, these VEGFR2 positive EPCs, only in our copper depleted patients. And as I mentioned, these are the cells that flip the angiogenic switch. Now I have to explain a little bit more on this one. So the other thing we looked at, so after we saw our results with the VEGFR2 positive EPCs, we said, wow, that's interesting, but are we doing anything else? And being incredibly, I don't want to say cheap, conservative, I said, I'm not going to go into my banked samples until I really know what I'm looking for. So in oncology, we call it a fishing expedition. I'm not doing it. I'm too old to do a fishing expedition. So we actually went back, set up another series of experiments in which we had a mouse model again, where we gave some of the mice TM and half of them we didn't. And then what we did is that we looked in the lungs because we wanted to see what happened to the premetastatic niche. And so what we found in the TM-treated mice, that we totally took out this enzyme called lysoloxidase in the TM-treated mice. And we also found that because we did that, that affected collagen scaffolding. Now, collagen scaffolding is really important because, you know, when tumor cells go and land someplace, they have to land somewhere. They land on a collagen scaffolding. So it's almost like a grid that they land on. We found that we totally messed that up. So we said, this looks like it's worthwhile to go back to our banked patient samples in order to see, do we do anything to lysoloxidase? And thank goodness I didn't waste my specimens. And you can see that over time, we decreased lysoloxidase only in the copper-depleted patients. These lysoloxidase is really critical, and there have been a number of reports that have come out literally over the past couple of months how absolutely critical it is for metastasis in probably triple-negative breast cancer. They talk about ER-negative breast cancer. So this is really important, and lysoloxidase is really heating up right now. So I was happy that we had that. So it's great you have a drug that might be doing something, maybe, at least on the biologic endpoints. We haven't talked about outcome in patients yet. But, side effects, people don't want side effects. And luckily, this drug is very well tolerated. So in terms of, what do we care about? Well, first thing, we don't want to harm people. But when we talk about side effects, we have a grading system. And when we talk about grade 3, 4, those are severe side effects. So in over 2,000 cycles that we've given, we've actually caused neutropenia in 3.2% of patients. And then neutropenia basically means they're over copper depleted. And I would tell you that about half of those, we over copper depleted because we found we could more efficiently copper deplete them. We gave them another pill, an antacid, and we probably we didn't realize we had to cut back. It's a learning curve, okay? We didn't realize we had to cut back on the TM. So we have a 3.2% neutropenia rate and only 1 febrile neutropenia. Febrile neutropenia is sort of important because you never want someone to get a fever if they have low blood count. They can get really sick. I would say that the most trying thing about this drug is the fact that you need to take it anywhere from 3 to 4 times a day. So it's well tolerated. So let's talk a little bit about outcome. In my world, when we talk about did someone's cancer come back, we call that progression-free survival. So when we look at the progression-free survival for all patients in the trial, it's 81%. But there's something that I want to point out to you guys. You may see this already, but you're going to see this in every curve I show you. And that is if someone gets it to this point, basically it seems like nothing ever happens. So this means no relapse. In here means relapse, okay? So it flattens out. So this includes all patients in the trial. So these are the stage 3s, and these are the stage 4 NEDs. When we look at the adjuvants, the patients who have stage 2 and stage 3 breast cancer, the progression-free survival is 91%. And again, you see this flattening out. When you look at the stage 4 NEDs, again, you see a flattening out. When we look at it by molecular subtype, remember I told you the triple negatives, that's a particularly aggressive molecular subtype. You can see there's no difference. That's the red line here. And again, you see a flattening out. And when you explode out the triple negatives, you can see that for the stage 4 NEDs, the 71%, flattens out. And for the adjuvants, it's 91%. And I will tell you that, and I don't have this graph up here, that for when we look in the adjuvant population that we have, there's no difference between triple negative and non-triple negative. And that is not normally what you see. So perhaps we're doing something. Overall survival is 86%. And it's a median follow-up of 4.9 years. So it's what would be considered in this population more than adequate follow-up in order to see if there was an effect, you should see it by this time. Okay, so what I'm going to do, you're probably saying, oh, those graphs, Kaplan-Myers, what does that really mean? And I want to put it into context for you. But I want to put it with a certain caveat. When you're a clinical investigator, you can go down a slippery slope if you start using historical controls. But I feel just to sort of give you a sense of why we're excited about it, I'm going to show you these slides. But just remember, slippery slope, historical controls. So my fellow, we presented this data on Monday at ASCO, and usually when you have the honor of giving an oral session, they have a discussant. Somebody puts it into context. And one of my colleagues, Mark Mawasser, who is a HER3 guy, we call him, down at UCSF, was the discussant at the session. And he kindly lent me his slides. So they're not my slides. It's his opinion, but I feel like he is so good on graphics that I think it'll probably convey to you why we're excited about this. Okay, so this proposal tests. So what TM does is what we believe is it's keeping micromets. And the way that we think about cancer is that there's tumor burden, and this is time, and you have surgery, chemotherapy, and it basically reduces tumor burden below subclinical. To a subclinical level. But at some point, what happens is that it starts to grow, and then you have a recurrence. And so what Mark thought, and this is really nice graphics, is that by giving TM for 2 years, that perhaps you push the recurrence all the way out. So keeping subclinical occult disease, subclinical and a cunt. So he thought that perhaps there was a signal of efficacy, and there might be. And just sort of, again, putting it into context, this is the PFS of the triple-negative patients by stage, and these are the stage 4 NEDs. You see that it flattens out at about about 2 years, and it's 71% disease-free. And so how does that compare to other contemporary clinical trials in the same population? Well, there are 2 trials that Mark has actually compared it to. So this is a phase 3 trial of a PARP inhibitor, eniparib, in gem-carbo with patients with stage 4 triple-negative breast cancer. And as you can see, the median survival for stage 4 triple-negative breast cancer is less than a year. Okay, so this is what you normally see, and this is what our trial shows. Furthermore, what Mark did, and hopefully I can do this right, is that he basically changed the axis so that you could really see, squeeze down to superimpose, and you'll see what the normal is, and you see what we are. So we're excited. Looking at another, but that's one trial. Looking at another trial of a gemcitabine taxane, Gem-Cis versus Gem-Taxol, 2 chemotherapy regimens for stage 4 triple-negative breast cancer. Again, using a squeezed axis to superimpose on our 7-year axis there. You can see that median survival, as what's published, is less than a year for stage 4 triple-negative breast cancer, and we're at 71%. So this is why we're excited. So what about the adjuvants? Right, adjuvants are always a little trickier. In our high-risk triple-negative population, this is normally what you see. You see basically for the same time frame about 50%. There are other studies that show basically for our population it's down at about 40%, a large MD Anderson data set. He brings in another context slide, and you can see it's about 50%. So it's encouraging. And I think that was it. So what can we say about all this? Well, I mean, certainly TM is safe and well tolerated. There's no doubt about that. We believe that copper depletion is affecting some of the biological processes within the tumor microenvironment that is known to promote tumor metastasis. We have a lot of things which are being evaluated right now back home in the lab. We're looking at circulating tumor DNA in order to see, well, what happens to it? Does it go away after a certain amount of time? Are we affecting tumor dormancy? We're looking at other copper-dependent biologic processes in order to see, are they affected by TM? Probably going to look at some of the matrix metalloproteinases, SDF1, things like that, in order to really sort of create a story. What processes are we affecting? What's most exciting to us is that tumor occurrence after 2 years is an absolutely rare event. If people get past 2 years, it appears that they do not relapse. And as clinical oncologists, that's really exciting. So where are we going with this? Well, I am working my butt off to try to get a phase 3 trial going. So hopefully that'll work. So is this disruptive? I hope so. Stay tuned. And the end. Thank you. So with that, I can take some questions. Yes?
Early on, you talked about multiplying out that $295,000 number. Would there be, I know pricing is complicated in pharmaceuticals, but would there be an expectation that higher volumes, that there'd be pressures somewhere on payers to bring the price per dose down?
No, and I will tell you, this was really shocking to me. So if you have access to the ASCO website, and you can pull up Len Saltz's talk, he actually addresses that question exactly. So you would certainly think if there were more drugs in the market that there would be a downward price pressure. And in fact, it's the opposite is true. So for example, with CML, chronic myelogenous leukemia, a drug called Gleevec or imatinib was one of the first drugs that came out. That was literally a pill you could take to cure your leukemia. And so they were the first to market. I think it was Novartis, first to market. There was a certain price point. Then there were 2 other drugs, at least 2 other drugs that came up. All the drugs, pricing going up. So now it's about 3 times what it actually debuted at. So, and there are at least 3 drugs on the market. So it's all a question of what the market will bear. That was the take-home message from that talk. Yes?
So I just want to understand, when you gave the TM, these were like to a stage 3 or stage 4 triple negative.
And non-triple negative too. And non-triple negative.
Yeah, okay, but let's say with the triple negative, so had the tumor already gone to a point of regression, or what if you gave it to people that had that tumor, it was metastasized, and it had not regressed?
Yeah. So some people have done that. There was a study that was done out of MD Anderson in which they combined it with chemotherapy. And what they found is when they added TM, it made the chemo work better. And there are a couple of groups that are specifically looking at that in ovarian cancer, because one of the drugs we use in ovarian cancer, they think there's mechanistically that it helps it. It looks like it might be very important in certain subtypes of melanoma. And so the group at Duke, I've been talking to the group at Duke, because I know how to get TM, I help all these people. I'm like the TM queen, who knew? Basically, they're combining it with some of the BRAF inhibitors in melanoma. I think it slows things down, but I think if you really want to try to cure someone, I think you really need to get rid of any bit of cancer that you can see, and then you can start to pull out the infrastructure. Otherwise, I think you're slowing things down, which is not bad either, but it's not, I think, where the real value of this approach is.
Do you know what chemo that's
Well, they've combined it with carboplatin, and I think the melanoma guys are going to do it with trametinib. I've never used those drugs because I'm not a melanoma doc, but it's, if you're going to, in cancer, these days we think about lung cancer, breast cancer, melanoma, brain cancer, but the bottom line is, guaranteed within the next few years, you're not going to be thinking about cancer that way. You're going to think about it as, a BRAF mutated tumor or HER2 mutated tumor. The lines between cancer, how we classify it is totally blurring. And to reflect that, they have these thing called basket trials at the NCI, where you have a particular mutation, you get a drug for that mutation. Yes?
Thank you, Linda. You've been doing all these clinical trials from within Cornell. Very often, these usually are spun out into companies that are funded by venture funds, and still you're almost at phase 3 without any outside financing. How do you fund all this?
Lots of different ways. So being in New York and being at Cornell, there is a real culture of philanthropy. We got our first funding was from Breast Cancer Alliance of Greenwich, where we got, I think, not a lot, $60,000, but it was enough to sort of get us going. We got a large grant from Komen in 2000 and I'm going to say 2006 or so, just about before we started. Cancer Research and Treatment, New York Community Trust, patients, anonymous donors. I have not, and I really need to do, I think I'm afraid to look to see how much money I've spent so far. I probably have spent close to $1 million to get this far, I'm guessing, but it's all been, I always say it's, born from Peter to pay Paul, so we've been able to get it done.
1 million to phase 3.
1 million to phase 3.
And you're aware of how much it costs usually to go to phase 3 when you're a venture company, right?
Yes. Well, the thing is, what you have to understand is that we have an incredibly committed team. I have tight control, and we've been able to get it done. And I think that sort of reflects, I don't know why everything costs so much. I mean, we've been able to get it done.
You have two disruptive things then. First is
right.
the itself.
Right. Get it done.
Phase 3 at $1 million.
Yeah.
The cost of
I can tell you my salary, it certainly didn't go to my salary, okay? It certainly didn't. But, we have an incredibly committed team, and we have a lot of, just to give you an idea, I had the last lady who went on the trial, she came from and by the way, we have people who come to this trial from like all over the world. The last lady, she was from Newport Beach, and so I said, I'm always worried about money. And so she said, I'm interested in your TM trial, and I'm like, well, you'd be a really good candidate, but we stopped accrual, and we stopped. You see, we didn't go to 80 because I'm worried about money because we keep on rolling people over. And I said, but you can get it off trial. I'm more than happy to help you. And she said, something, I really want to help support your research. Turns out they have like a huge fund in Newport Beach, and she's been like tossing me money like since then. I mean, she's given me a little under $200,000 so far. So you never know where your money's going to come from, right? But I'm very tight on it because I'm afraid I'm going to run it. So one thing I didn't tell you is that, after the second patient enrolled on the trial, she was a triple negative stage 4 NED. We at 2 years, because we picked 2 years was an arbitrary number based on the available data at the time, for a biologic therapy. We saw that her cancer hadn't come back. We said, pretty interesting. We don't know, that could be normal. It could be what we're doing. We don't know, but let's continue her. So we continued her. At 4 years, her cancer still hadn't come back. So we were like, wow, okay, we wouldn't expect that because median survival for stage 4 triple negative breast cancer is less than a year. So maybe we might be doing something. So at that point, what we did is that we continued to write a series of extension studies in 2-year increments to further study this phenomenon. So we have patients who are on study on copper depletion for out to 8 years. We have a series of extension studies. We're up to 4. And we're hoping that the circulating tumor DNA that we're running now will give us some insight into when can they come off of it. Because, I didn't show you the data, but I think that there's a period of time during which tumors can metastasize. And if you don't sort of get it done in that tumor, in that period, I think the signal might go away. And that's sort of looking at through the lens of bone marrow-derived progenitors. Because don't forget, we collect a lot of data.
So we'll have one more question.
Yes?
Is TM cross the blood-brain barrier?
I will tell you that I think it probably does. I've never seen any studies, but I will tell you, in triple negative, we have one lady. She had brain mets when she first came in, got it taken out, and she hasn't had subsequent brain mets. So I think it probably does, but I think it's an effect that can take a while. But there are no data, as far as I'm aware, of specifically answering that question. Testing the CSF, seeing if there's any.
Is TM commercially available?
There's a black market in TM. So I buy it from Sigma Aldrich, and our investigational pharmacies compound it. There's a commercially available drug called penicillamine, which has a lot of side effects, which is used for Wilson's disease, which is available, as well as another drug called trientine, which is also commercially available. There is a company called Wilson Therapeutics, which has a long-acting TM, which is the one that I would like to move forward with the phase 3 trial. But if not, I'm going to go to NCI and get my TM drug repurposed from Merck. So just want to move forward.
Thank you very much.
Anyway, okay.